Bismillahirrahmanirrahim. In today's lecture, we'll be discussing the second important component of hemostasis, that is the coagulation cascade, which is also called as coagulation pathway. As you all know that coagulation of blood, it is also called as blood clotting. Simply speaking, it is a process by which blood changes from liquid to a gelatinous form, ultimately resulting in the formation of a blood clot. And this blood clot, it then results in cessation of the blood flow from the damaged blood vessel. When the blood clot is formed, it is ultimately followed by the repair mechanism, resulting in the hemostasis. Coagulation cascade, it includes a series of reactions that leads to deposition of an insoluble fibrin meshwork around the platelets and other cells. Your book has given a very good example. I usually quote it in my lectures that the coagulation cascade, it can be considered just like a dance in which one coagulation factor is passed from one partner to the next partner. This whole coagulation cascade, it involves various substances. These important substances, they include enzymes, a substrate, and a cofactor. The enzyme is the activated coagulation factor. Substrate is the inactive form of that coagulation factor. And the cofactor, as you all know, it is the reaction accelerator. Coagulation cascade is divided into two pathways. One is extrinsic and the second is intrinsic pathway. As in the previous lecture, I told you about the prothrombin time and the partial thromboplastin time. The prothrombin time, it is associated with the extrinsic pathway, while the partial thromboplastin time, it is associated with intrinsic pathway. Before discussing these two pathways, first I will tell you that which clotting factors they are associated with prothrombin time and which clotting factors they are associated with partial thromboplastin time. Please make a list because this is a very important question when you consider short essays or when you consider small parts of the auspice. Please note down the prothrombin time. It assesses the function of factor 2, factor 5, factor 7 and factor 10. Along with these four factors, it also assesses the function of fibrinogen. So prothrombin time, it assesses the function of five factors, factor 2, 5, 7, 10 and fibrinogen, while the partial thromboplastin time, it assesses the function of factor 2, factor 5, fibrinogen, factor 10. These were the same factors which are also assessed by prothrombin time. The additional factors which are also assessed by partial thromboplastin time, they are factor 8, factor 9, 11 and 12. So if we make a list of the factors that are assessed by partial thromboplastin time, they are factor 2, 5, 8, 9, 10, 11, 12 and fibrinogen. Now let's discuss these pathways, the extrinsic pathway and the intrinsic pathway. We'll start from the extrinsic pathway. And extrinsic pathway, it is activated when there is a damage to the blood vessel, resulting in the secretion of the factor 7. As you can see on the top right corner, there is a tissue injury resulting in the secretion of the factor 7. This factor 7, it comes in contact with the tissue factor. This tissue factor, it is also called as thromboplastin. When these two factors, they come in contact, it results in the formation of a complex that is called as tissue factor factor 7 complex. This complex, it then activates factor 9 and factor 10. So as you can see, the tissue factor and factor 7 complex is formed. It results in the activation of the factor 9 and factor 10. You can trace it in the center of your slide and towards the left side of your slide. So this activation of factor 9 and factor 10, it is a key towards the process of coagulation. Factor 7 is also activated by thrombin and few other factors like factor 9, factor 10 and factor 12. Once the tissue factor and factor 7 complex is formed, it is immediately inhibited by the tissue factor pathway inhibitors, factor 10 and its cofactor. It forms a complex which then leads to the activation of prothrombin to the thrombin. You can trace it downwards in the slide. There is activation of prothrombin, which is also factor 2, and resulting in the formation of thrombin, which is activated form of the prothrombin. This thrombin, it then activates the coagulation components like factor 5, factor 8, and when it is activated, it may result in the activation of more factor 9 and more factor 10, so that the cycle it continues. Now, if we look at the intrinsic pathway, it begins with the formation of the primary complex on the collagen by high molecular weight kininogen and precalciferin. It also includes calciferin and factor 12. This factor 12, it is also called as Hagman factor. High molecular weight kininogen, it is denoted as HMWK. Initially in the intrinsic pathway, the precalciferin, it is converted into calciferin, and this calciferin, it results in the activation of the factor 12. The activated factor 12, it leads to activation of factor 11, and factor 11 ultimately it leads to activation of factor 9 and also results in the activation of factor 8. This complex factor 8 and factor 9, it then results in the activation of factor 10, and hence this cycle it goes on. So intrinsic pathway, it is more straight pathway as compared to the extrinsic pathway. It starts with the activation of the precalciferin to the calciferin. Calciferin results in the activation of factor 12 or the Hagman factor. This activated factor 12, it leads to activation of factor 11. Factor 11 causes the activation of factor 9 and factor 8, and this factor 8 and factor 9 complex, it leads to activation of more factor 10, and hence the cycle it goes on. Moving further, let's discuss the functions or the important functions of the thrombin. Thrombin is the most important factor. It causes conversion of the soluble fibrinogen into the insoluble form, that is called as fibrin. And as you people have studied in the previous two lectures of hemostasis and the platelets, that fibrin, it creates a meshwork around the platelets and the other cells. The second important function of the thrombin is the activation of the platelets through protease activating receptor. It was also told in the previous lecture of platelets. It has few other functions like it has pro-inflammatory effects, and it also have anticoagulant effect to some extent. So this is a picture that denotes the few functions of the thrombin. As previously I told you, the four important functions, the top of the list being the activation of fibrinogen to insoluble fibrin form. There are various cofactors which are also involved in the coagulation pathways, and these cofactors, they include the calcium, they include phospholipids and vitamin K. Calcium and phospholipids, they are important for the formation of the prothrombin complex, as well as vitamin K is also essential for adding of the carboxyl group to the glutamic acid residues of factor 2, factor 7 and factor 10. Factor 9 is also included in the list. So, briefly speaking, calcium and the phospholipids, it is important for the formation of the prothrombin complex. While vitamin K, it is essential for adding up of carboxyl group residues to the glutamic acid of factor 2, factor 7, 9, and 10. So these are the important cofactors that take part in this coagulation cascade. The cofactors, there are few regulators which are also associated with the coagulation pathway or coagulation cascade, and they are protein C and protein S. The activated forms of these proteins, protein C and protein S, they degrade the factor 5 and factor 8. If there is deficiency of any of these regulators, it may result in the thrombotic state, because as you know that factor 5 and factor 8, they are procoagulant. So if their function goes unchecked, it may result in a thrombotic episodes in the body. So those thrombotic episodes, they are prevented with the help of these two regulators, protein C and protein S. And if there is deficiency of any of these regulators, it will result in the prothrombotic state. Now we have studied the coagulation cascade, and once the coagulation in the blood vessel it has started, it must be stopped at the site of the vascular injury. Otherwise, it may carry some harmful effects. It may lead to the thrombosis and ultimately ischemia. So how this coagulation it should be limited? One very important step that Allah Pak has created, one very important regulatory step that is present in our body is the washout effect of the flowing blood. This blood flow, it washes out the activated coagulation factors, and those activated coagulation factors, they are ultimately removed by the liver. So this is one counter regulatory mechanism or a rate limiting step of the coagulation pathway. The second important pathway, that is the fibrinolytic pathway. So as the coagulation cascade is going on side by side, there is also a fibrinolytic pathway that is operating. This fibrinolytic pathway, it limits the size of the clot, and later on it results in the dissolution of the clot. And how this pathway works, it is shown in the next slide. This slide it shows the fibrinolytic pathway, and this fibrinolytic pathway, it mainly involves the plasmin. Plasmin is produced in the inactive form called plasminogen in the liver. Plasminogen itself cannot cleave that much fibrin, so it should be activated to the plasmin for the fibrinolysis to occur. Two important activators of the plasminogen, they are the tissue plasminogen activator and urokinase. These are the agents that convert plasminogen to the active plasmin, so allowing the fibrinolysis to occur. Tissue plasminogen activator is released in the blood very slowly by the damaged endothelial cells, such that after several days, when the bleeding has stopped, the clot is broken down. This occurs because plasminogen, it becomes entrapped within the clot when it is formed. As it is activated slowly, it breaks down the fibrin meshwork. Tissue plasminogen activator and the urokinase, they are also activated, and they can result in the activation of more and more plasminogen. So their function, they should also be inhibited. So this tissue plasminogen activator and urokinase, they are themselves, they are inhibited by plasminogen activator inhibitor, as denoted in the slide, and it is abbreviated as PAI. In contrast, the plasmin, it further stimulates the plasmin generation by producing more active forms of tissue plasminogen activator and urokinase. Now plasmin, as it is present, it has become activated. The plasminogen, it has become activated into plasmin or the free plasmin. This free plasmin, it can lead to further fibrinolysis of the blood. So this free plasmin, it should also be inactivated, and this free plasmin, it is inactivated with the help of alpha 2 plasmin. So this alpha 2 plasmin, it is very important in activating further plasmin molecules. As a result, that fibrinolysin, it does not progresses. So this was a lecture about coagulation cascade and the fibrinolytic pathway. In the next lecture, we'll be discussing the third important component of the hemostasis, that is the endothelium, and alongside that we'll start the various hemorrhagic disorders which we need to study in the hemodynamics. So till then, take care and Allah Hafiz.